Bismillahirrahmanirrahim. Welcome. This is Biochemistry 2, Chem Code 342. Today we are having lecture 14, and our today's topic is transferase enzymes. This is Dr. Naeem Khan, Associate Professor, Department of Chemistry, CUST. In today's lecture, we will talk about transferase enzymes description. Then we will discuss the important types of transferase enzymes. And then we will study the important reactions which are catalyzed by the transferase enzymes. And especially we will refer our example to those reactions which are very much important in the in the metabolic pathways. So let's have a start. We know that as per the International Union of Biochemistry, we have 6 major classes of enzymes, and these include oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. It means this transferase enzymes is one of the 6 major classes as per the classification of the classification recommended by the International Union of Biochemistry. So it is the second class of the 6 major classes of the enzymes. Before that, in our last lecture, we have discussed in detail the oxidoreductase enzymes. We know that these are the enzymes which are actually catalyzing the oxidation reduction reactions. I mean redox reactions, and which are very much important in the metabolic pathways of the living things. Out of the oxidoreductase enzymes, we were having major classes. One of them was hydroxylases. And we have discussed that these are the enzymes which catalyze the addition of hydroxyl group to their substrates. And we have studied their chemical reaction too. Next class was the oxidases. This is the oxidoreductase enzyme which we studied that it is catalyzing hydrogen or electron acceptance by oxygen in their substrates. I mean, this class includes the enzymes which catalyze such reactions in which involve the acceptance of hydrogen or electron by the oxygen in their substrates. Third class was the peroxidases. And the peroxidase are the oxidoreductase enzyme which catalyze the reduction of hydrogen peroxides. Next class of the oxidoreductases we already studied was reductases. And the reductase enzymes are those which are catalyzing the reduction reaction, means addition of hydrogen or electron or removal of oxygen is called reduction. Next, we already studied one of the oxidoreductase enzyme. Subclass was oxygenases, and oxygenases are the oxidoreductase enzyme which catalyze the incorporation of oxygen into their substrates. And the sixth and the last major subclass of the oxidoreductase enzyme which we studied was dehydrogenases. And dehydrogenase, as the name indicates, these are the enzyme catalyzes oxidation by dehydrogenation. I mean, removal of hydrogen from their substrate. So these 6 classes of the oxidoreductase enzyme, which is the one of the 6 classes of the enzyme, class number 1, we already studied. And we know that oxidoreductase enzymes are the enzyme catalyzing oxidation reduction enzyme reaction, and these reactions may be hydroxylases, oxidases, and peroxidases, reductases, and oxygenases, dehydrogenases. So today, let's to have a start of the transferases. Here we can see transferase enzymes are the enzymes which are catalyzing the transfer of a group of atoms between a pair of substrates. It means these are the enzymes which are catalyzing such reactions involving two substrates. And these enzymes catalyze the transfer of a group from one substrate to another substrate. Let we have substrate S1 and the enzyme is transferring a group of atoms, which may be anything, to a substrate S2. A group of atoms may include, let's suppose, maybe phosphate group. This may be include any acyl group, for example, acetyl group, or including any group. We will discuss the major groups which are transferred the transferase enzyme one by one in the types of transferase enzyme. But one thing is clear, the transferase enzymes are the enzyme which will catalyze the transfer of a group of atoms from one substrate to another substrate. For example, if we have substrate S1 and S2 like this, substrate S1 is having a group represented by G, and if this reaction is taking place and this group G is transferred to S2, in this reaction the group G, which may be anything, is transferred from substrate S1 to substrate S2. Such reaction is catalyzed by the definitely enzyme, for example, because the biochemical reaction are catalyzed by specific enzyme. So any enzyme which is catalyzing such reaction will be called a substrate enzyme. Once again, transferase enzymes are the enzyme catalyzing a group of atoms from one substrate S1 to substrate S2. For example, if we have substrate S1 and if a group G is transferred to another substrate S2, such a reaction is called catalyzed by the enzyme, and the enzyme are known as transferase enzyme. This class of enzyme include those one which are catalyzing the transfer of, for example, amino group. Aminotransferases are very much important enzyme. We know, especially in the biosynthesis of amino acids, we know that if non-essential amino acids are lavishly available in the body, the essential amino acid can be even synthesized from them. And this is only possible through definite biochemical reaction. Also, we know keto and amine transferase reaction, especially in the Krebs cycle, where the keto group is transferred to amino group is transferred to keto group. So such reactions are important. Transferase enzyme include the transfer of amino group. Also, they include the transfer of phosphate group. It may be transferring the acyl group, especially we know about the acetyl group, which is transferred. So these transferase enzyme may be those one involving the transfer of amino group, phosphate group, acyl group, or may be glucose group, because glucose is a very important part of our diet and it is involved in the so many biochemical biosynthetic reaction. So these are etc. These four are the main groups which are whose transfers, for example, the transfer of amino group, phosphoryl group, acyl group, and glycosyl groups are the important groups which are actually catalyzed by the transferase enzyme. And these are transferred from substrate 1 to substrate 2, and such reactions are catalyzed by the enzyme known as transferase enzyme. Going to the types of the transferase enzyme, as in the last lecture we discussed, there are four important groups which are actually transferred by the transferase enzyme. On the basis of these important groups, we have 4 types, and one type is the transaminases. Transaminase. Trans means transferring, aminase means NH2 group, amino group. Transaminase, as the name indicates, these are the enzyme which catalyze the transfer of amino group from one amino acid to a keto acid. There are so many keto acid in the body, alpha ketoglutarate, very important in the Krebs cycle. And amino acid, yes, amino acids are the building blocks of proteins. So in the body, there are so many amino acids are available. So transaminase is the type of transferase enzyme which catalyze the transfer of NH2 group amino group from amino acid to a keto acid. For example, here we can see this picture is from my thesis. So here, this is glutamic acid, L-glutamic acid. Look, this is alpha carbon in which amino group is attached and also acid group is attached. So it means an amino acid. And we know the basics amino acids. This is aspartic acid. We have one CH3 and glutamic acid. We have two CH3 and then double carboxylic acid group are present. This is amino acid double carboxylic group having, which is known as glutamic acid. In this reaction, this amino group is transferred to this acid. What is this? This is a very important compound known as pyruvic acid, and this is synthesized in the glycolysis. We know that in the glycolysis from one glucose we get 2,3 carbon compound known as aspartic acid. But remember, this is a keto acid because the functional group here is a ketone. Because there is a one carbon on one side, other carbon on the other side, and here we have carbonyl functional group. Such functional group is called keto group. And as we have acid, so this is keto acid. In this reaction, we can see in the product alpha ketoglutarate acid, and we get another amino acid, amino group, carboxylic group, and methyl group. This amino acid is known as alanine. If we look into this reaction, glutamic acid react with pyruvic acid, and we get glutaric acid and alanine. So what happened? This NH2 highlighted as red is transferred to this group, and this functional group is transferred to here, glutamic acid. What we get? This is actually functional group exchange reaction. Amino group is transferred from the glutamic acid to the aspartic acid, and in return, functional group, carbonyl functional group is transferred to the glutamic acid. We get alpha ketoglutarate and alanine. Alanine is an amino acid. This is actually in the second amino acid after glycine. It is one of the 20 amino acids we know. And this ketoglutarate or ketoglutaric acid is the product of reaction 5 in the Krebs cycle. So we know that this is also very important residue, intermediate, that is required in the Krebs cycle. Such reaction in which amino group is transferred from amino acid, this is amino acid, to a keto acid, this is a keto acid, and in return, amino acid is transferred to keto acid and keto acid is transferred to amino acid. These are transferase enzyme because they are transferring a group, and such transferase enzyme which are catalyzing the transfer of amino group are called transaminase enzyme. Next type is the kinase enzyme. Kinase are very important enzyme which are actually, whenever there's a kinase word, so we come to know something in our mind about the phosphorylation, dephosphorylation, or addition of phosphate, something like that. These are the enzymes which are catalyzing the transfer of phosphoryl group, phosphate group. And phosphorylation and dephosphorylation is actually, we can say, energy storing and energy releasing process. If we consider the electron transport chain. And also if we remind that in the glycolysis, actually the first reaction is actually the phosphorylation. That glucose gets phosphorylated and we get glucose 6-phosphate. So kinase enzymes are important ones. These are the transferase enzymes which are transferring the phosphate group from one substrate to another. Look, this reaction is also from my thesis. This is adenosine triphosphate. Adenosine, this is, look, this is adenine, this is ribose sugar, and we have three phosphate attached through phosphate group. This is adenosine triphosphate. The highlighted red phosphate is the third phosphate, which is actually energy rich. And we know that actually this phosphate is the one which is actually transferred in the energy. And in this reaction, we can see here that the other substrate is the glucose. Glucose pyranose. Pyranose form glucose structure is given. Here, the phosphate group is transferred from here to this carbon number 6 of glucose. We get glucose 6 phosphate, means phosphate is linked to the glucose 6 carbon. And when one phosphate is removed, it means that ATP, adenosine triphosphate, is converted to adenosine diphosphate. One phosphate from ATP is actually transferred to the glucose, and we get from glucose to glucose 6 phosphate. In this reaction, what happened? There is a transfer of phosphate from ATP, one substrate, to glucose, another substrate. Such enzymes, which are catalyzing the transfer of a group like phosphate from one substrate to another, will be called as transferase enzyme. And this type of transferase enzyme, which are catalyzing the for transfer of phosphate, are called as kinases. Remember, in this reaction, we know that the enzyme involved is the hexokinase, is the journal name, or if the substrate is a specific glucose, so glucokinase is more specific enzyme which is catalyzing in this enzyme, in this reaction. So whether it is a hexokinase or glucokinase, but the important thing is a transferase enzyme catalyzing the transfer of phosphate from one substrate to another. Such enzyme catalyzing the transfer of phosphate from one substrate to another is called transferase, and this subtype is kinase. So after first type of aminotransferase, in the last lecture we discussed transaminase, which was transferring amino group. No, the next type is the kinase, which is catalyzing the transfer of phosphate group. So they are called kinases. Third type of the transferase enzymes are those which are transferring the transfer of, which are catalyzing the transfer of glucose residue, glucose glycosyl residue from one molecule to another. Usually, we know that in the glycogen synthesis, there is a primer that is 4OH usually of remaining glucose, or we know that in the glycogen, there is a primer which if all stored glycogen is consumed, then the protein serine OH will be used. But anyway, there is a residue of glycogen remaining, which is known as primer, that is actually used for the linking of glucose. So such enzymes which are catalyzing the transfer of glucose from one species to another will be called glycosyltransferase. And this glycosyltransferase, in example I'm giving is those one which are transferring glucose residue one by one to the glycogen primer, and in this way glycogen is synthesized. Glycogen is a storage form of glucose that is in the liver and also in the muscle. Mostly that is stored form in the liver, and whenever there is a need, so glycogen is broken down, or if there is a high glucose, glycogen is synthesized. The basic reaction behind is this one. There is a glycosyl transfer, one by one glycosyl is transferred, and that is converted to glycogen. Look at this reaction. This is also from my reaction. Look, this reaction, this is uridine diphosphate glucose. UDP here with the UDP, uridine diphosphate. Uridine structure is not given because that's somewhat a big structure. So just a name is given, uridine. Along with the uridine, this is one phosphate, this is another phosphate. So uridine diphosphate, that is linked to glucose. Glucose carbon number 1 is linked to with the UDP. So we have uridine diphosphate glucose. In the glycogen synthesis, biosynthesis of glycogen, this compound is synthesized, and whenever it is synthesized, then it is brought to the glycogen synthesizing end. This is a primer. Primer is usually the remaining glycogen part, or it may be protein if the glycogen is completely exhausted. But normally this is a remaining glycogen. Here this exposed end is available, and whenever UDP glucose is reacted to glucose with the glycogen primer, here the glycosyl residue is transferred and it is combined here with the carbon number 4 of the attached primer end, growing end, you can say growing end. Now in the product we can see that this is glycogen is length is extended by one glycosyl unit. This was glycogen primer, and the glycosyl component is um, catalyzed. It's transferred by the glycosyltransferase enzyme, and now the glucose unit is linked to the growing end of the glycogen. Now we have primer with whose length is 1 unit increase, and we get uridine diphosphate. The glucose unit is released. This uridine diphosphate will again go, it will be glycosiding, it will be attaching glucose with the glucose 6 phosphate with the hexokinase enzyme, and then the kinase enzyme will undo when after glycosyl units attached will come here, and they will be attached here, and the glycosyl unit will be transferred. Such transferase enzyme which are catalyzing the transfer of glucose group from one molecule to another are called transferase enzyme, and they are named specifically glycosyltransferases. And these are very important in the biochemical pathways, as we can see that glycogen synthesis it is involved, and glycogen is very much important because it is helping to maintain the glucose level constant in the body. Another type of transferase enzyme is the acyltransferases, acyl group. This is alkyl carbonyl group link. This is called acyl group. It may be acetyl if this R is CH3. These are the transferase enzymes which are catalyzing the transfer of acyl group. So if acyl group from one substrate is transferred to another one, such will be called as acyltransferases. For example, see in the reaction. Here we have one of the reactant is choline. Choline is very important. We know that from the lipids point of view, they are a very important component of the compound lipid, compound lipid, especially lecithin, cephalin, and plasmalogen. This choline we know. When it is reacted to the acetyl coenzyme A, actually coenzyme A structure is not given, it's a very big structure, but we know that coenzyme is linked through sulfur and then acetyl group here. CH3 C double bond O, this is acetyl. Acetyl coenzyme A is been reacted with choline by the choline acetyl transferase enzyme, and we get there the acetyl choline. Look, this group is actually this group is linked. This acetyl group is now linked with the choline, and this hydrogen is captured by the coenzyme. So coenzyme A transfers acetyl group to choline. So now from choline we get choline, acetylcholine. Okay. So what happened in this reaction? In this reaction, actually, acetyl group is transferred from acetyl coenzyme A to choline. Such reaction in which acetyl group or acetyl group is a type of acyl group that is transferred from one substrate to another are called acyltransferases or acyltransferases, and this is one of the important types of transferase enzymes too. Again, Western Todd book is a very important book, and GL Jain, the Indian, that's also recommended books. Please have a thorough, detailed study of these topics we studied today, and highlight the important points. Add to this. This may not be the end. Please extend your study. And from today, thank you. Allah Hafiz.